All right, so let's move on from the cell wall and talk about some other components of the prokaryotic cell. So we have the cytoplasmic membrane, or the plasma membrane. So I've already kind of showed you the construction of the membrane. We talked about the phospholipids that make up the membrane. So it says a bilayer, lipid bilayer, so that's your phospholipids with proteins embedded. So the proteins help to determine kind of how that membrane functions. All right, so even though the cells are separated from their environment by a cell wall and a plasma membrane, they still have to interact with their environment to obtain nutrients, maintain water balance, exchange gases, and to dispose of waste products. So we typically describe a membrane as being selectively permeable. It's going to allow certain materials to move across the membrane so that these materials can move in and out of the cell, so that the waste can move out of the cell and the nutrients can move in. It also says energy reactions, because if you think about it, a membrane helps to separate two areas, and we can create concentration gradients across that membrane, and that can help us to convert energy into different forms. Now, within the confines of the plasma membrane, we find the internal structures of the cell are bathed in a watery cytoplasm. So it says 70 to 80% is water that makes up the cytoplasm. So prokaryotes lack membrane-bound organelles, so there's very little compartmentalization that occurs within the cytoplasm. So most of the biochemical reactions are going to happen here. We can have inclusions or inclusion granules. These are basically used for storage. They're not considered to be organelles because they're not membrane bound. They're basically distinct collections of substances inside the cell. So usually it's some types of granules that won't dissolve in water. So the bacteria tend to build up some of the substances in times of excess. You got plenty of it, so we'll just kind of stow it off to the side here. And that's what you're seeing in this image in the lower right. These kind of large circular structures are inclusion granules. And so this happens a lot of times when bacteria is grown in culture, when it's grown on a very nutrient-rich media. So it has kind of all the nutrients it needs, so it'll stockpile them in inclusion granules and then simply break them down when they need them. We also have the cytoskeleton. So this is kind of a skeletal framework that provides structure and support for the cell. It's typically made of very long protein filaments. Those filaments that are part of the cytoskeleton can assist with the construction of cell walls. They can help when the cell gets ready to divide. But typically the cytoskeleton composition is different from the cytoskeleton of eukaryotic cells. It's not quite made of the same types of molecules, but it does help to support the overall shape of the cell, kind of given an internal framework. Now, the genetic material for prokaryotes is DNA, and the DNA is organized into a single circular strand that's located in an area that's called the nucleoid. So this is just a region. It's not an organelle, right? Because if it was, we would call it a nucleus, and it would be bound by a membrane. But remember, our prokaryotic cells lack membrane-bound organelles. That means they do not have a nucleus, but it doesn't mean they lack DNA. They do have DNA. It's just located within a region that's called the nucleoid. Okay, so that's where we find our circular DNA strand. We may also find additional pieces of DNA. These are called plasmids, and these are small circular strands of DNA as well. Plasmids also tend to be double-stranded, and they can be taken in during conjugation when we have that pili that extends between two bacterial cells. So a plasmid can move from one cell to another cell. And this is typically how antibiotic resistance is transferred from one bacteria to another is through a plasmid that's exchanged between them, or plasmids can be taken up through the pores in the membranes as well. So the plasmids, the key about them is that it's DNA that contains genes that are not central to the functioning of the cell, right? The DNA that's crucial for the functioning of the cell is in the nucleoid region. The plasmids are just extra DNA that they can exchange between other cells. So all cells must must make proteins to survive. And prokaryotic ribosomes are the location where proteins are produced. Now, keep in mind, we said prokaryotes lack membrane-bound organelles. Ribosomes are an organelle, but they're not comprised of a membrane. They don't have a membrane that surrounds them. They're mainly comprised of ribosomal RNA. So these ribosomes that we find inside of prokaryotes are different than the ribosomes that we find inside eukaryotes. So prokaryotic ribosomes have a lower mass and diameter than eukaryotic ribosomes. And because of this, we can use antibiotics to target them, and they won't target eukaryotic ribosomes, right? So we can differentiate between the prokaryotic and the eukaryotic ribosomes based upon what we call the sedimentation rate, which is basically how fast the particles in a solution settle out when they're spun in a centrifuge. So a centrifuge is basically like your washing machine on the spin cycle, right? Centrifuge is going to spin solutions at very high speeds, and whatever is very heavy can kind of fall out of solution and will appear at the bottom of the tube that it's in. So we measure the sedimentation rate using what we call Svedberg units. So that's what the S stands for when we're looking at this 30S or 50S is the Svedberg units. So prokaryotic ribosomes, just like eukaryotic ribosomes, are made up of a large subunit and a small subunit. So the small subunit for prokaryotes is what we call a 30S, so that's its sedimentation rate, and the large subunit is a 50S. And when the subunits combine together to complete the ribosome, you would normally think, okay, 50 plus 30 would be 80, but that's not quite the case. Our full ribosome is is a 70S instead. So what happens is when the large and the smaller subunit come together, they kind of change a little bit of how the molecules interact with each other, changes the shape of those two subunits, and it kind of gives us a different sedimentation rate. So total is 70S for bacteria. The total ribosomal sedimentation rate is 70S. If we compare this to eukaryotic ribosomes, they have an 80S. All right, so eukaryotic ribosomes are comprised of a large subunit, which is 60S, and a small subunit, which is 40S, but the two combined together give you an 80S ribosome. The archaea, their ribosomes are also 70S, just like our bacteria, but they're actually constructed and resemble more like a eukaryotic ribosome, so they're a little bit in between there. Now, interestingly, if you think about this, the chloroplast and the mitochondria of eukaryotic cells, these are organelles, membrane-bound organelles that we find in eukaryotic cells. They actually have organelles inside of them. They have ribosomes inside of them that are 70S. So the chloroplast and the mitochondria of eukaryotic cells have a 70S ribosomes in them. So what do you think this hints at if they have 70S ribosomes inside of them? I'm going to let you guys stew on that for a little while. And we'll talk about that with eukaryotic cells. Some bacteria make endospores to survive very harsh conditions. The endospore are basically inactive structures. They're dormant, and they allow the cell to just kind of survive in hospitable environments. So bacterial spores can resist a lot of environmental stresses, like if there's not enough nutrients, if it's too hot or too cold, or it's too dry, or even radiation or various chemicals, they can survive those conditions. There have even been reports that scientists have been able to grow bacteria from spores that were 250 million years old. So what happens is, here we've got a bacterial cell, and at one end of the cell it will start to form a spore. So endospores are not reproductive structures. They simply produce another more resistant structure inside of them, and then eventually the outer the cell of the bacteria degrades or lyses and dies, and this spore is what is left, and that can then survive the harsh conditions. And when conditions change, they germinate out and produce another bacterial cell. So it's not reproductive. They don't make more and more and more bacteria. They simply allow that single cell to survive in hospitable conditions. So we got a concept checked here. So it says, where in a bacterial cell would you find the genetic material? Do you find it in the nucleus, the nucleolus, the nucleocapsid, or the nucleoid? So notice that they all sound very familiar. All right, so nucleus is in eukaryotic cells. A nucleolus is also in eukaryotic cells. A nucleocapsid we haven't really talked about yet. All right, kind of referring to another group that we haven't talked about, but it is the nucleoid. We said the nucleoid region is where we find the genetic information, the DNA of a bacterial cell. All right, last but not least, I wanted to mention briefly about the archaea and how they differ from our bacteria that we have been talking about. So we've talked about the shapes of bacteria. So archaea also have spherical rods, spirals, as far as their shape goes, and they can have other irregular shapes. They do have single circular DNA. We mentioned them having the S layer more so than regular bacteria. And protein synthesis, though, is very similar to how eukaryotes go about protein synthesis using their ribosomes. We said the ribosome is a 70S, just like bacteria, but the structure resembles more like eukaryotic ribosomes. They do not have peptidoglycan in their cell walls. Instead, they have the pseudopeptidoglycan, and their cell walls have much more diverse construction than we see in the bacteria. If we look at their evolutionary relationships with the other domains, they are actually more closely related to the domain eukarya than they are to the domain bacteria. So even though they're prokaryotes, just like the bacteria, they show a more recent common ancestor with the eukarya. And for the most part, a lot of times we refer to archaea as extremophiles, that they live in very extreme environments where a lot of other organisms may not be able to survive. But we also know that we are starting to look for them in other locations. We just haven't really had the means to detect them. So we do know that some archaeans actually live on the human body. But once again, we have not been able to contribute any type of diseases to the presence of the archaeans.